What makes T-cell lymphoma hard to diagnose? T-cell lymphoma poses unique challenges when it comes to diagnosis. One of the main reasons is that it includes over 20 different subtypes, each with its own characteristics. These subtypes often share clinical and microscopic features, making it tricky to tell them apart from other lymphoid cancers or even benign conditions. A significant hurdle is the lack of specific molecular markers. Unlike some cancers that have unique identifiers, T-cell lymphomas generally do not have distinct markers that can confirm their presence. Many of these subtypes express similar proteins on their surface, which complicates the classification process. Tests like flow cytometry or immunohistochemistry, which look for these proteins, may not provide clear answers. Under the microscope, T-cell lymphoma cells can look very similar to cells from other lymphomas or even normal lymphoid tissue. This overlap requires skilled pathologists to interpret the results accurately. Often, additional tests are necessary to reach a definitive diagnosis. The classification of T-cell lymphomas is complex. The World Health Organization has identified multiple subtypes, such as nodal T follicular helper cell lymphomas and angioimmunoblastic T-cell lymphoma. Each subtype has its own set of markers and genetic features, which can be quite subtle. This means that a comprehensive panel of tests, including gene expression profiling, may be needed for an accurate diagnosis. Another factor is the genetic and immunophenotypic variability among T-cell lymphomas. Some subtypes, like T-cell prolymphocytic leukemia, have specific genetic abnormalities, but these are not always present. Therefore, doctors often relying on a combination of clinical findings, genetic information, and immunophenotypic results to make a diagnosis. T-cell lymphomas can also present in unusual locations outside of lymph nodes. They might appear in the skin, gastrointestinal tract, or bone marrow, which can mimic other diseases and delay diagnosis. This extranodal involvement adds another layer of complexity. To accurately diagnose T-cell lymphoma, a multidisciplinary approach is often required. This means that hematologists, pathologists, and oncologists must work together, integrating clinical information, imaging results, and various test outcomes. In summary, diagnosing T-cell lymphoma is challenging due to its biological diversity, the absence of exclusive markers, and overlapping features with other conditions. Accurate diagnosis is crucial because treatment options and prognoses can vary widely among the different subtypes. If you suspect you or someone you know may have T-cell lymphoma, it is essential to seek evaluation from specialized centers that have expertise in lymphoma diagnosis and management.